Good afternoon, everyone, and welcome to today's topic. We are going to talk about emerging therapies for inherited retinal diseases. My name is Katie Frederick. I am the Digital Content Manager for Vision Aware, part of the APH Connect Center. I'm going to now pause the recording and we will begin with our ACV REP credit information. And again, welcome everyone to today's webinar on the topic of emerging trends in inherited retinal diseases. And we are pleased to have Michelle Glaze and her colleague Ben joining us from the Foundation Fighting Blindness. And Michelle is the Director of Professional Outreach at the Foundation Fighting Blindness, and we are pleased to have her and Ben with us today to discuss this topic. And Michelle, I will hand it over to you.
Great, thank you so much. Welcome everyone, and thank you to team at American Printing House for allowing us the opportunity to share some information with your group. I enjoyed presenting back in February, and we're excited to be able to share again with you and look forward to other opportunities to collaborate in the future. So as mentioned, I'm Michelle Glaze, Director of Professional Outreach for Foundation Fighting Blindness. And about 20 years ago, I was diagnosed with an inherited retinal disease called retinitis pigmentosa. I was very much surprised by the diagnosis. No one in my family has an inherited retinal disease. So I didn't really know what to think, where to turn, or what my future would look like. After doing some research, I stumbled upon Foundation Fighting Blindness. And for the first time, I was just really interested about all that was ongoing in the way of research. It provided hope for me for the future. And so I'm excited today to be able to turn things over to Ben Shaberman in a little bit to share with you all of the wonderful clinical research that's underway. Before doing so, I just want to share a bit about the Foundation Fighting Blindness. The organization has been around for over 50 years. The foundation is the world's largest private funder of retinal degenerative disease research. And the organization's mission is to drive research to find treatments and cures for blinding retinal diseases like retinitis pigmentosa, Stargardt disease, Usher syndrome, LCA, and even forms of dry age-related macular degeneration. Over the last 50 plus years, the foundation has driven a variety of different types of research projects, lab, translational research, clinical trials, natural history studies. The foundation also funds a no-cost genetic testing program called the MyRetina Tracker Open Access Program. And the foundation also funds some career development awards for up-and-coming research in the field of inherited retinal disease. Through the work the foundation does, we really seek to provide the gifts of knowledge, community, and hope for patients with an inherited retinal disease or retinal degenerative disease, as well as for their families. During today's webinar, you're going to learn about a lot of clinical research from Ben Shaberman. He's the Vice President of Science Communications. Ben has been with the foundation for nearly 20 years. He has studied, reported, educated, and followed clinical research in the inherited retinal disease field. He is the host of the Foundation's Eye on the Cure podcast, in which much of this research is showcased. And Ben has a knack for sharing this very detailed scientific information with individuals like myself who are, going through a journey with vision loss. He does it in a way that's very simplistic and easy to follow and understand. So I am excited to turn over the presentation to Ben Shaberman. And Ben, take it away.
Well, thanks, Michelle, Katie, and all of you associated with American Printing House. So glad you could join us and spend a little time learning about inherited retinal diseases and some of the research underway to overcome these conditions. And that's basically going to be the flow of my talk. I'm going to give a quick intro to the retina. We're going to talk about some of the more prevalent of these rare diseases, and then talk about some of the research underway. So the first slide that I have up is a side view of the retina, and it shows the many different cellular layers. Sometimes we think of the retina as a layer cake of different cells. And amazingly, this is, of course, blown up considerably, but the retina is only a half a millimeter thick. It lines the back of our eyes like wallpaper or tissue paper. And what the retina does is it processes light. When light comes in through the front of our eye, it goes through the middle of the eye, through the vitreous to the back of the eye, and it's the retina that takes that light and converts it into electrical information that's sent out through the back, through the optic nerve to the back of the brain to the visual cortex. And that's how vision occurs. The retina is really in partnership with the brain to make vision possible. Now, at the top of this diagram, excuse me, are longer cells. And these we call photoreceptors, or rods and cones. And these cells are the cells that actually process light and create those electrical signals. And amazingly, each of our retinas has 125 million of these rods and cones. Now, concentrated in the center of the retina, in what we call the macular region, are the cones. And in this diagram, they're colored red, green, and blue. Cones give us central vision. They give us the ability to read and recognize faces. They help us perceive colors. They're the cells we use for vision when we're in a lit setting, a lighted setting. Rods, on the other hand, are concentrated on the periphery of the retina, and rods give us peripheral vision and night vision. And that's basically the layout of the photoreceptors. Now, there are other cells in the retina that perform important functions, but it's only the rods and cones that are naturally light sensitive. And on the next slide, I have an image of an actual, and I believe this may be a human retina. And this is captured using an electron microscope. And these longer kind of tubular cells, thin tubular cells that are lavender in color, these are rods, very beautiful in this picture. And then nestled within the rods are cones. They're shorter, kind of stubbier and darker. They're almost magenta. And this is just what an actual retina looks like, again, captured using an electron microscope, which is not something your typical eye doctor, not even a typical researcher, has access to that kind of magnification. But it's a beautiful image, and I wanted to share it and describe it to help you appreciate the beauty of the retina. Another thing that I think is really amazing about the retina is that it processes more oxygen for its size than any other part of the body. More than the heart, the lungs, the brain. Again, this is for its size. It's a real workhorse. And while during the day, it's processing light to help us see, it also goes through a regeneration process when we wake up in the morning. The tips of our rods and cones are sloughed off, they're shed, and they're disposed of, and those tips regrow. So right when we wake up in the morning, our retina is going through this regenerative process so that we have new fresh tips on our photoreceptors to process light. So that's a quick introduction to the retina. I wanted to now get into some of the retinal conditions that the foundation is concerned with. And the first category of conditions is what we call the inherited retinal diseases. And collectively, these inherited diseases affect about 200,000 people in the US, and that extrapolates to more than 4 million people worldwide. And these are conditions, many of which Michelle mentioned: retinitis pigmentosa, Stargardt disease, Usher syndrome, choroideremia, LCA. And I'll talk more specifically about those, at least some of those, in a minute. But many of these conditions are diagnosed in childhood. That's when they start causing vision loss, and they often, almost always, progress as people get older. Vision continues to decline. And in a very, just to talk about progression generally, a lot of people with these conditions are legally blind by the time they're 40 or they reach middle age. Variation in these diseases can be quite broad. Some people have more severe disease. Some people have more moderate disease. But in general, legal blindness is fairly common in middle age. The thing that's common also to all these conditions is that for every person affected, they have one gene that's mutated that's causing their diseases. So we call these conditions monogenic. The challenge is it could be one of more than 280 genes that can be the culprit. If you put 5, 10, 15 people with an inherited retinal disease in a room, most of them will have different genes causing, different mutated genes causing their conditions. And it really underscores the need for genetic testing so that we understand specifically which mutated gene is causing a person's vision loss, is causing their disease. So the inherited retinal diseases collectively are rare. Again, they affect about 200,000 people in the US. Another condition, age-related macular degeneration, is different in a few different ways. First of all, it's very prevalent. It affects more than 10 million people in the US and is in fact a leading cause of blindness in people over 55 years of age. And while there are genetic factors that can increase the risk of AMD, aging, unfortunately, we're all getting older, aging is the most significant risk factor. Aging and also smoking. Smoking, having a history of smoking, increases the risk of AMD. So in terms of the way AMD occurs, it's not all that different from other conditions like heart disease, cancer, etc. Our risk increases as we get older, and lifestyle factors like smoking can increase our risk. And so the foundation, as Michelle mentioned, we're targeting the inherited retinal diseases, and we're also targeting dry AMD because of the two forms of AMD, wet and dry. We only have one FDA-approved treatment for dry, while there are many for wet. Now, one last general comment I wanted to make about the retina is that the retina is neural tissue. It's actually an extension of the brain. Ultimately, it's connected to our brains through the optic nerve. And that makes it an excellent therapeutic target for researchers who are trying to understand and develop therapies for the neural system, whether it's the brain, the central nervous system. The retina is really the ideal place. And there are a few reasons why the retina is ideal for evaluating neuronal therapies. The retina is a small piece of tissue, so you don't need a lot of therapy to test it in the retina. It's relatively accessible. While we don't normally like to think about things being put in our eyes, the eye is a lot more accessible than the brain or other parts of the central nervous system. And also, we have two retinas. So in a clinical trial, if you're trying to understand a therapeutic effect on the retina, you can treat one eye, treat one retina, and compare it to the untreated eye or the untreated retina to see if that therapy is having an effect. So a lot of cutting edge therapies have been developed, or I should say are being developed, with the retina as the proving ground. So I want to quickly review some of the more common of the inherited retinal diseases. Of the 200,000 people affected by these conditions, retinitis pigmentosa is by far the most common. It affects about 100,000 people in the US, so about half of all people with inherited retinal diseases. It is a condition that's usually diagnosed in childhood. In some cases, people with less severe disease may be a little older, but again, most people are in childhood or adolescence. Retinitis pigmentosa usually affects rods first. So people have night vision and peripheral vision issues. And over time, as people lose rods, vision constricts. It becomes tunnel-like. And as the disease gets worse, it can be like looking through a paper towel holder, and even more, in more advanced cases, it can be like looking through a straw. And for some people, vision can constrict to the point where there's only a pinhole left or actually nothing left. Again, they can vary in their severity, and there are about 80 genes, each of which, when mutated, can cause retinitis pigmentosa. It can be inherited in a number of different ways, recessively, where parents are unaffected carriers of the child that's born with the disease. It can be a dominant disease where a parent can have the condition and have a 50% chance of passing it on to their son or daughter, or it can be X-linked. And X-linked inheritance gets a little complicated, but what usually happens is women with X-linked disease are unaffected, but they're carriers, and their sons are at risk of inheriting the gene and then actually having the disease. And so on this slide, I have what we call fundus photos. This is what an eye doctor sees when they look at the back of your eye through the pupil. They see the retina, and the retina, the healthy retina, has this nice orange reddish glow. There's a yellow spot toward the left on this diagram. That's just the optic nerve. The color of the retina gets a little deeper toward the center where the macula is and the fovea is. That's where there's a greater concentration of those cones that give us central vision. And then on the right, I have another fundus photo, but this is the image of somebody who has retinitis pigmentosa. And on the periphery of the retina are dark clumps, and that shows areas of degeneration. And again, this is what an eye doctor sees when they're looking at the back of the eye, and they can more obviously tell that something is happening in this retina, and the clumping on the periphery is a hallmark feature of retinitis pigmentosa. So another fairly common inherited retinal disease is a condition called Usher syndrome. It affects about 25,000 people in the US. And essentially, Usher syndrome is RP, but people with Usher syndrome also have hearing loss, and they have problems with their vestibular system. They have problems with balance. And Usher syndrome can be severe, it can be moderate, and there are some rare cases with Usher syndrome type 3 where the variation can vary a little bit. But in Usher syndrome type 1, people are born with profound hearing loss, And then they're usually diagnosed with RP in childhood, and then vision starts to decline. In Usher syndrome type 2, hearing loss is less severe, it's moderate, and usually hearing aids are adequate to help people gain some usable hearing. But again, RP is diagnosed in childhood or a little later in life. And then Usher syndrome type 3, which is more rare, can vary in its severity and onset. There are more than a dozen genes, each of which can cause Usher syndrome when those genes are mutated. And Usher syndrome is almost always inherited recessively. So parents are unaffected carriers, and they have a 25% chance of passing each of their mutated genes onto the son or daughter who will, again, get the condition. Now, I'm not showing any fundus photos for Usher syndrome because this condition is basically, in terms of the retinal onset, exactly the same as RP. And then finally, one other relatively common inherited retinal condition is called Stargardt disease. This is an inherited form of macular degeneration. So unlike Usher syndrome and RP, which cause outer retinal degeneration and peripheral vision loss that constricts over time, people with Stargardt disease lose central vision first. And they develop this blind spot, or we call it a scotoma, that increases over time. And in relatively early in this condition, people begin losing their ability to read and recognize faces. And Stargardt disease is caused by the accumulation of toxins in the retina that cause that central degeneration, that central vision loss. And unlike Usher syndrome and RP, where there are multiple genes that, when mutated, can cause the disease, most people with Stargardt disease have mutations in the gene ABCA4. Again, it usually is diagnosed in adolescence or young adulthood, but I have met people even in their 40s with much less severe disease who are diagnosed and genetically tested to show that they have Stargardt disease. Now, about 30,000 people in the US have Stargardt disease. And this is, again, fundus photos. On the left is that healthy retina with the orange reddish glow. On the right is the retina with a lot of yellow flecks or yellow spots. They're especially concentrated in the center of the retina. Those are waste products known as lipofuscin. These are the deposits, those waste products that accumulate and cause the degeneration of the retinal cells. That's the hallmark of Stargardt disease. Now, there are many other retinal diseases. I'm just going to breeze through these very quickly for the sake of time. One is called choroideremia. It's not that dissimilar from RP, but it also affects other parts of the retina, not just the photoreceptors. It affects a supportive layer of cells called RPE cells, and it affects a layer of vasculature called the choroid. But it causes basically the same kind of symptoms. Retinoschisis is a splitting of the retinal layers. It's X-linked in many cases, meaning boys are affected, women are the carriers, the moms are the carriers. LCA, Leber congenital amarosis, is simply a family of conditions that causes significant vision loss at birth. Kids are born with relatively severe vision loss. LCA, again, really a family of conditions. There are more than 24 genes associated with LCA. Achromatopsia is kind of the inverse of a lot of retinal conditions. Instead of causing night blindness, achromatopsia causes day blindness. People have problems seeing in lighted settings, And in fact, bright light can be painful. There are 5 genes that, when mutated, can cause achromatopsia. And then Best disease and cone rod dystrophy are inherited forms of macular degeneration. And there are more. So there's a lot of diversity within that group of 200,000 inherited retinal disease. I hope this gives you a sense of some of the more common ones and some of the others that can cause vision loss. So now I'm going to switch gears and talk about some of the research underway to try to save and preserve vision caused by these conditions. And we're at a very exciting juncture in our history. As Michelle mentioned, we've been around for more than 50 years, and it's really over the last dozen or so years that we've had this tremendous surge in human research. We've been able to launch clinical trials for emerging therapies. And currently, there are somewhere in the neighborhood of 45 clinical trials underway for emerging therapies. It's a very exciting time. And I'm proud to say that the foundation has played a big role in getting many of these therapies into the clinical trials. We don't necessarily fund all of these 40 clinical trials, but we funded the research that made them possible, that got these therapies that to the point where they were ready for a clinical trial. And specifically, we funded about 88% of the current IRD therapies that are in these human studies. And excitingly, thanks to the research we're funding, we're attracting outside investments from companies who are working to move these in clinical trials and across the finish line. And on this slide, I have listed 39 companies that are currently in our space that are focused on inherited retinal diseases. Some have clinical trials underway, others are working toward launching clinical trials. But it's really exciting to see the level of commercial activity that's occurring, because you need these investments to get these treatments through the later stage clinical trials and out to the people who want the therapies. By far and away, the most exciting advancement in our space is FDA approval of a gene therapy called Luxturna. And so when I talk about gene therapy, what I mean is replacing the mutated gene that's causing somebody's disease with a healthy copy. So remember, I mentioned there can be anywhere, there are about 280 genes, each of which, when mutated, can cause a retinal disease. Well, we're working to try to replace some of those genes to help people, and that's what gene therapy is all about. So Luxturna is specifically designed for people with mutations in a gene called RPE65. People with mutations in the gene often get leber congenital amarosis. In some cases, they get RP. And in early clinical trials for this particular therapy, it had a dramatic effect for people who had these forms of retinal disease. It enabled people to put away their white canes. These folks that were treated could see the faces of their loved ones for their first time. Some were even able to see stars in the sky or fireflies. So this gene therapy, Luxturna, again for people with RPE65 mutations, provided a real proof of concept for researchers showing that gene therapy was in fact an effective way to treat these conditions. And as this therapy moved through a clinical trial, the clinical trial sponsor at the time, Children's Hospital of Philadelphia, spun off a company called Spark Therapeutics to move this treatment across the finish line. And now, per the previous slide that I showed you, a lot of other companies are getting into this space. So we're excited about the promise of gene therapy, especially gene replacement therapy, but you're going to see also some gene agnostic approaches that are showing promise as well. And when I say gene agnostic, I mean therapies that are designed to work regardless of the mutated gene causing the retinal disease. But next, I wanted to review some exciting progress that's been made in gene replacement for people with a retinal disease called X-linked retinitis pigmentosa. This form of RP affects about 20,000 people in the US and in the EU. It's usually caused by mutations in a gene called RPGR, and we have 3 companies that have clinical trials underway for this form of gene replacement therapy. MiraGTx is in phase 3. They had vision improvements in their earlier phase. If phase 3 is successful, they will likely seek FDA approval for their gene therapy. AGTC is in phase 2. They as well have shown vision improvements. They're moving toward phase 3. They'll likely start their phase 3 trial in 2024. And then a third company, 4D Molecular Therapeutics, is still in phase 1/2, and we're waiting to hear more results from them. So a lot of exciting work going on for X-linked RP. But now I want to shift gears and start talking about some of those gene agnostic therapies. Again, therapies that are designed to work regardless of the mutated gene. And one company that just received authorization to launch a clinical trial, they are called Sparing Vision. They're developing what we call a neuroprotective gene therapy. Their gene therapy produces proteins, specifically a protein called rod-derived cone viability factor, that's designed to keep cone cells healthy and functioning, regardless of what the gene is that's causing the disease. So we call this neuroprotective. It's injected underneath the retina, and one injection is designed to last for many, many years, just like other gene therapies. This is for people with RP, Usher syndrome, and possibly some other related retinal conditions. This will begin at the University of Pittsburgh Medical Center and also in Paris, France. Sparing Vision happens to be a French company. And we've been funding this work for decades at the Institut de la Vision in Paris. And now we're funding the company, Sparing Vision, helping them to advance this and actually some other therapies that Sparing Vision has in their portfolio. So again, gene agnostic, neuroprotective, trying to keep those cones healthy, because cones give us central vision, detailed vision, and the ability to see in lighted settings. Now, another approach that's very exciting is for people with advanced retinal disease, advanced RP, advanced AMD. And this is designed to restore light sensitivity to people who have lost all their rods and cones. Now, on the right side of this slide, I have that side view image of the retina that I showed earlier. But I have a big ominous X going through the rods and cones. And this is to signify somebody who's lost all their rods and cones to a retinal disease like RP or AMD. Now, because they've lost all their rods and cones, any therapy to preserve the rods and cones or to replace the mutated gene or to again, save rods and cones before they degenerate, those therapies aren't going to help because these folks have lost all their rods and cones. But what researchers realized is that there are other cells in the retina that survive after rods and cones are lost. And I have those circled on this diagram. These cells are known as bipolar and ganglion cells. These cells normally don't process light. They have other functions in the retina. But what researchers have done is they've delivered either a gene or a chemical to these cells to bestow them with light sensitivity. So in essence, they're using these alternate cells, these bipolar cells and ganglion cells, as a backup system for people who have lost all their rods and cones. And excitingly, there are 4 companies in clinical trials for this approach. And we call this approach optogenetics. And all of these companies, there are different stages of clinical research, but all have shown that they can restore at least some basic light sensitivity to the retina. And specifically with nanoscope and bionic sight, they've enabled people to better navigate, in the nanoscope case, a mobility course. In the bionic sight case, there are anecdotes where people could see, for instance, one gentleman could see his dog running through the snow after getting a treatment. Another person could see candles during the holidays. With the higher dose of the bionic sight therapy, people could identify vegetables and the different symbols on playing cards, hearts, diamonds, spades, clubs, etc. So very encouraging results for people who really had very little, if no vision left. Now, as these approaches move through the clinic, we're hoping we see even better results. Will this restore 20/20 vision? I don't think we're at that stage yet. Perhaps some more advanced approaches down the road will do that. But for now, the approaches that are in the clinic now show promise for restoring some rudimentary vision to people who have, again, lost most or all of their vision. Another exciting gene agnostic approach is this powerful antioxidant developed by Nacuity Pharmaceuticals. They're in a clinical trial in Australia. What researchers observed in lab studies and natural history studies is that oxidative stress plays a big role in exacerbating vision loss in people and animals with retinal disease. So they studied antioxidants in animals to see if using an antioxidant could in fact slow vision loss. And they saw that it did in fact do that. These antioxidants did that. They studied one specific antioxidant called N-acetylcysteine. They modified that N-acetylcysteine. They added an amide molecule to it to make it, an amide to it to make it more bioavailable and more potent for the retina. And then they moved it into a clinical trial again in Australia for people with Usher syndrome. This is an oral medication. Again, it's designed to work regardless of the mutated gene causing the disease. And they're hoping that in late summer, early fall, they will get some readouts from the trial. If results are encouraging, they will launch a site for this trial in the US. Now, Johns Hopkins, along with the National Eye Institute, is studying the original molecule that was studied in the lab, N-acetylcysteine, and they're actually launching a phase 3 trial for it as well in the United States and some other countries in Europe and other parts of the globe as well. So the modified, more potent molecule is in a phase 2 trial now in Australia. The original molecule is moving into phase 3, and we're hoping that at least one of these is successful. This is appropriate for somebody who, again, still has some rods and cones left, and they're trying to save their vision and slow the degeneration. So last, I wanted to talk about a cell therapy for age-related macular degeneration that recently moved into a clinical trial. So this, again, is a cell therapy. In people with age-related macular degeneration, they lose a layer of cells called RPE cells. RPE cells are like a support system for photoreceptors. When you're looking at those layers of the retina in that earlier diagram I showed, the RPE are like a blanket of cells that the photoreceptors are nestled in. And RPE are critical to the survival and the health and well-being of photoreceptors. When people lose RPE, their photoreceptors will ultimately degenerate as well. Now, in people with age-related macular degeneration, they lose RPE cells, and again, their photoreceptors degenerate as well. So the idea is to transplant new RPE cells to prevent the degeneration of the photoreceptors. Now, what's really cool about the way the RPE cells are developed for this particular therapy is the researcher in this trial, his name is Kapil Bharti from the National Eye Institute. He's taking blood cells from patients, just a small sample of blood, and he's tweaking those cells so they revert back to a stem cell-like state. So they become stem cells, and as stem cells, they can be coaxed to become virtually any cell type in the body. They're, again, a primitive cell type. They're a clean slate. So then his lab is coaxing these to become RPE cells. So essentially, the patient is their own RPE cell donor. The blood cells are extracted from the patient. Again, it's just a small sample of blood, and those cells can be made into RPE cells. He's putting them on a scaffold, this biodegradable scaffold, so they're properly oriented into that sheet-like structure. The scaffold also helps the cells integrate and survive. And this trial is still at a relatively early stage, so they haven't reported results yet. But what's real exciting is the process that I just described to make these RPE cells. We call this inducing pluripotent stem cells. And because the donor of the cells is the patient, we don't have to worry about immunogenicity. We don't have to worry about immunosuppressing the patient because the cells are coming from another person or an outside source. So a great advantage of this approach. And this is the first use of induced pluripotent stem cells for the retina in the United States. And we're hoping and we're confident that researchers will use this approach for other forms of retinal disease in the not too distant future. We are funding other researchers who are developing therapies using induced pluripotent stem cells for other retinal conditions. And so I covered a lot of ground in the past, 40 or so minutes, an intro to the retina, some of the diseases, some of the exciting research. If you want to learn more, whether it's about the conditions, the retina, or the research, I encourage you to go to fightingblindness.org. And now I think we have about 15 minutes for questions. And I will open up the chat here to see what people are asking about.
If you have a question, do feel free to put it in the chat. It should be enabled for everyone,
Okay,
or
and
at
I'm
least hosts
noticing,
and panelists. So
and I'm noticing there's a lot of good information in the chat about like how to access us, our website, but there are no specific questions. So if you want to unmute, or I don't know, Katie, are people able to unmute?
yes,
Okay.
we do have some hands.
Okay. Yeah, I guess it makes sense to raise your hand first.
Yes.
So we can call on you.
All right. All right. So the first one is Christopher. So you should be able to unmute.
Hello, thank you for allowing me to talk. Can you hear me okay?
Yes.
Yes. Hi, Christopher.
I want to tell you that the chat is disabled.
Okay, thank you. I will try to fix that. It's working on our end, but I'm sorry that it's not working. So I will go back in and change that. We're able to chat with everyone, so I don't know. Maybe try again. I know I changed the setting, but I will go back in and change it back. All right. All right. In the meantime, let's go back to the hands here. We will do hands, that will work. All right. Aaron. Whoops.
Hello.
Yes, we hear you.
Hello.
Hi, how are you?
Good.
Curious about any kind of development on treatments for achromatopsia.
Sure. Good question. So Mira GTX, a company that's partnered with Janssen, has a couple of trials underway for achromatopsia caused by mutations in CNGA3. Two different therapies, one for CNGA3 and one for B3. And they've reported some preliminary encouraging results. And I know they're working to move those forward. So I don't have a lot of detail because the results that they reported were relatively early, but they're working to move those two gene therapies forward.
Thank you.
Sure.
All right, William.
Yes, thank you. Can you hear me okay?
Yes.
Yeah.
Excellent presentation. Can you tell if there's any research that's going on with Batten's disease?
Sure. And for our participants who aren't aware, Batten disease actually can vary in its severity, but it can be a pretty difficult condition and cause serious problems with many different organs and bodily systems. But there is a gene, or I guess it's either a gene therapy or a protein therapy in the clinic for people with mutations in the gene CLN2. And I apologize, I think it might, I'm not remembering, but it's an injected therapy. I think it's injected into either the brain or another part of the central nervous system to deal with Batten's disease.
Okay, thank you. I'm an educator, and so what has happened with us is often we find children who are identified with RP initially, and then as the disease progresses, we see the speech and the motor and some of the other degenerative situations, but it mostly shows up for us initially as RP.
Right. And what I would encourage you to do, if it's doable for you, is to go to our website. I know we have an article on a therapy or two that's in development for Batten's. I know about the one for CLN too. There might be a second one as well. I just don't know off the top of my head.
Okay, thank you very much. I appreciate
Sure.
it.
And this is Michelle. That is a good reason for obtaining genetic testing, as early on as possible if there is an inherited retinal disease detected, just to make sure that clinical diagnosis is confirmed. So if anyone has questions about genetic testing, we certainly can help with that as well.
All right, Teresa.
Hi, I would like to know what clinical trials are being held in the Boston area.
Okay. I don't know offhand what's going on in Boston, but I can tell you that at Mass Eye and Ear, the lab that we fund there, the Birmingham lab, it's one of the best clinical research centers in the country, really in the world. And there's a website called clinicaltrials.gov. And what you can do is you can search by your condition, you can search by location or other keywords, and you can see what's going on at Mass Eye and Ear in Boston.
Thank you.
Sure.
All right. Sandra. You just need to unmute, Sandra. There you go. We don't hear you if you're speaking. All right, Sandra, you should be able to speak now. All right, we are not hearing you, unfortunately, so we will go on and hopefully you can get your audio issues resolved. Angie. You just need to unmute, Angie.
Okay, there. Can you hear
There
me?
you go. Yes.
All right, thank you so much. Thank you, Ben and Michelle, for wonderful presentation and wonderful education. I'm the president of the Indianapolis chapter for FFB, so love it when you guys speak and educate. Quick question, I have RP1. So with these gene agnostic theories, I know it's much easier to apply these, I'm assuming, to something that's recessive, or typically those are more easily, more clinical trials, rather than the dominant gene. They're a little trickier. So RP1 is dominant. So with these gene agnostic, would they also apply to dominant RP?
And Angie, I have a few things I want to say. First of all, you have a genetic cousin, so to speak. Maybe that's not the right word to use, cousin. Because Michelle has RP1 mutations, but she has recessive.
Oh,
Yes.
interesting.
So-
I didn't know that.
Yeah.
Okay.
Her case is a little more unusual because you're right. More people with RP1 mutations have dominant. And you're correct in that, without going into all the details, treating dominant disease can be a little more tricky if you're approaching it genetically, because often you have to knock down the mutated allele or the mutated copy in addition to replacing that mutated copy. But excitingly, at Mass Eye and Ear, they are developing, it's actually a CRISPR-Cas9 therapy for RP1, the dominant form. That is not in a clinical trial yet, But they're working to move that toward the clinic. But to get back to your question about the gene agnostic approaches, yes, all the sparing vision, the acuity, those all apply to you, and they're more gene agnostic. So yes, as somebody with RP1, if you have some vision remaining and you're trying to save what you have, those gene agnostic approaches definitely apply. And one last thing I wanted to say, we have a partner organization called Hope in Focus, and they are a group of families, most of whom are affected by LCA. And they do a lot of great work. They raise money for us, but they're holding their annual conference in Indianapolis on June 23rd and 24th. And my colleague, Chad Jackson, and I are moderating the scientific part of that conference. So
Wonderful.
I realize you have RP, but you could still learn a lot and meet other people in the area, again, most of whom are affected by LCA. If you want to send your contact information to Katie, I can send you more information about the conference. It's a wonderful conference.
Yeah, I would love that. Did you say
Thank
June
you.
23rd and 24th?
Correct.
Okay. Thank you so much. Katie, I will be emailing you.
All right. All right. We probably won't get to all the hands. And again, I apologize for the chat. I thought I had it fixed so everyone could chat. Sometimes closing the chat and then opening it helps as well. Sandra, are you able to speak? All right, let's go on. Juan. You just need to unmute, Juan.
Hello?
Yes, there you go. There you are.
Perfect. Hi, thank you. Thank you for the presentation. I just have a question in regards to the clinical trials. Is there anything in regards to LCA, specifically on the gene NMNAT1 or LCA9?
Yes, thanks, Juan, for that question. And actually, we're having this Boston theme come up again and again. So the lab at Mass Eye, they developed an NMNAT1 gene therapy, and a company that we helped launch called Opus Genetics has licensed that technology. And their plan is to ultimately move that into a clinical trial. I don't have any more details at this point. They have a pretty large portfolio of projects. NMNAT1 is one of them. And I would say stay tuned to the Foundation website for updates, for more information.
Thank you. And we do need to wrap this up, but I am putting my email in the chat. So feel free to email me with your questions, and we will get those answered in the manner that we can. Apologies that we, again, could not get to everyone's questions, and apologies as well for the chat not working as hoped. So technology is great when it works, right? So I want to again thank my colleagues Michelle and Ben for joining us today, and thank you for this great presentation. I will now stop the recording and provide the closing ACVRP credit code.